Think about it. B cells, armed with B cell receptors, can bind specific epitopes of the pathogen. This triggers that B cell to divide, to engage in a process of clonal expansion. Thousands of progeny, daughter cells, are produced exact copies of the mother cell. Each B cell produces antibodies that specifically correspond to the pathogen's epitope that stimulated the original B cell in the first place. Okay, but how, you may ask, does this help us fight infection? You surely recall that viruses are decorated with spike proteins, and that these spikes must bind to their receptors on the surface of the target cell in order to mediate cell entry. Only then is a virus actually able to infect us and begin to replicate. So now consider a B cell whose B cell receptor just happens to correspond to the viral spike epitope. And even more, specifically to precisely that part of the spike responsible for binding to its cell surface receptor. The encounter of such a B cell with its virus initiates clonal expansion, and now thousands of daughter cells begin to produce specific antibodies that target the viral spike protein. The viruses are attacked, first with IgM, but by the end of a few weeks of training, optimized IgG-guided missiles are produced. These latch onto the spikes, intercepting the viruses and blocking them from binding to the cells of our body. Binding of antibodies to spike proteins can neutralize the virus and prevent infectious disease from the very get-go. Thanks to immunological memory, should the same virus attempt to attack us a second time, the optimized, ready-to-go trained B cells stored in our memory ignite a powerful, focused response to knock out the virus before disease has a chance to set in. This ability to recall is the basis of vaccination, as we will soon learn. Now, to be honest, viruses are not wimps and have some tricks of their own, as we will also see in the next unit. Some viruses mutate and modify the structure of their epitopes. In this way, they effectively disguise themselves so to become less recognized by the antibodies that trained to bind the original viral features in the first place. Fortunately, antibodies have some flexibility in accommodating to change and variation in the targets they bind. Hence, an antibody designed and perfected to bind a specific epitope can cross-react and bind variations of that epitope, but pay a price of lesser efficiency. Nonetheless, cross-reactive binding is often enough to halt infection of a virus that is mutated. So you see, there is a dynamic dialogue between our immune system and the viruses they attack. B cells mount perfected antibody responses to block invading viruses. Viruses under attack accumulate mutations in an attempt to masquerade their epitopes and evade immune attack. Thanks to cross-reactivity, antibodies can nonetheless use first-generation antibodies to fend off evading viruses until a second round of antibody production takes over. Antibodies also assist in combating bacteria. As you recall, resident macrophages eat to defeat. Macrophages devour bacteria that invade our bodies. Some bacteria, however, tend to be slippery and hard to catch and hard to hold on to. If these bacteria, however, encounter B cells, they can launch clonal expansion and the production of antibodies specific for the slippery, coated bacterial surface. When antibodies bind and cover such bacteria with antibodies, the stalk effector signals are presented outward. These stalks are recognized by the macrophage, who has special effector signal receptors on its surface. Binding the antibody effector signals by macrophages gives them something to latch onto and increases their efficiency in eating. Coating a donut with chocolate sprinkles enhances our eating experience. So antibodies can neutralize viruses, prevent disease, and assist macrophages in dealing with bacteria. However, in all of these encounters, the pathogens, viruses, or bacteria interact directly with the B cell to stimulate clonal expansion and antibody production. But how does the immune system cope with viruses that have succeeded to infect our cells and begin to replicate? Here is where T cells make all the difference.